Before CRISPR, it cost someone $10,000 to make a single gene editing protein. But with CRISPR, the whole cost dropped to something like 10, even at most $100.
Feels like the hands of the god, that it can change the genes and the living forms. From playing video games in China's Silicon Valley to revolutionizing human biology, Professor Le Cong's journey wasn't planned.
I think I'm fundamentally driven by curiosity when I make decisions. I wanted to, in some way, maximize the sci-fi nature of my life. It's sometimes less about whether you are the smartest person to work on a scientific problem or startup. It's more like, do you really believe in it so you keep working on it?
But it was tragedy that set him on this path.
Both my grandparents on my mother's side passed away in the span of a couple of months. But when I asked around, no one seemed to really have a sense of why they passed away so quickly. Why we don't know how people get sick. What are the ways we can really help people? That's when I started to think about maybe I should take a career in medicine. Almost 2 years of my PhD work never got published. There are a lot of failures and probably more failure than success. But eventually, right, people mostly only see the success.
Now his breakthrough could reshape what it means to be human.
What if you can improve our ability to live better, healthier, longer? Maybe there's also a way to improve healthy living, to give ability to rejuvenate our stem cells for anti-aging purposes. We still have no cure for type 1 diabetes. There's still a very big question like what actually causes Alzheimer's? What causes aging?
But when you can rewrite human nature itself, should you? Today, my guest is Le Cong, an assistant professor at Stanford University School of Medicine, where he's pioneering the fusion of AI and gene editing to treat cancer and immune diseases. He's one of the co-inventors who proved CRISPR gene editing technology could work in humans, turning a $10,000 lab process into a $100 scalable technology. Professor Cong is a recipient of the NHGRI Genomic Innovator Award, Baxter Foundation Faculty Scholar, and named to Zhang's top 10 under 40. He previously served as a VP at Moderna, leading their gene editing innovation team, and has built a 20,000 plus member global community advancing AI for science. Professor Cong reveals the untold story behind CRISPR's breakthrough, from 2 years of failed experiments to the intense global race for discovery. He shares his counterintuitive philosophy of optimizing for curiosity over predictable outcomes, which led him from McKinsey to scientific discovery. We explore why he believes current medical progress is too slow despite successes like Ozempic, his vision for using AI to tackle diseases like Alzheimer's and diabetes, on CRISPR's 3 game-changing applications: curing inherited diseases, engineering better crops, and potentially extending human healthspan. Plus, why he thinks we're entering a new era where AI will accelerate scientific discovery at unprecedented scale. Let us get started. I want to start from the very beginning because I think your story is fascinating and there are many lessons people can learn from. I know you grew up in a place called Zhongguancun, which is also known as Chinese Silicon Valley.
Right.
How was the life there?
Thank you. Yeah. First, glad to be here. It's fascinating to talk about our work as well as our broader sort of interest in AI for science and biomedicine. I think, I basically, in some way, I did grow up in the equivalent of Silicon Valley, in at least for Beijing, right? I don't think there's probably a debate about whether Shenzhen or other places are also very exciting, but in Beijing, Zhongguancun was one of the first places where a lot of IT companies started. I was there. So maybe very quickly, I grew up in Zhongguancun playing computer games.
Wow, when was that?
That was probably 1992, 93.
That was like, you probably are the first group of kids in China who got access to computers.
And, we were getting, this, R-Valio, right? 286, right? 386 generation. To play very simple,
OG.
simple
OG
games.
For computers. Yeah.
And then maybe 3 examples, right? We were playing with these early computer games, and I think we were buying, for example, one of the first or second PCs from Legend, right, Lianxiang. And then the second example is at the time I remember we were setting up internet through Yinghaiwei, which is one of the early ISP provider of internet.
They put a sign in the highway how far away.
China is internet, right?
And then 500 meters, go to Yinghaiwei. I think that was a very phenomenal ads.
It's waiting for the modem to dial, right? And then so I was initially really pure text-based BBS, user at the time when I was still in elementary school. And the last part is, I think being, there were a lot of, when I walk to and from my home to school, there were a lot of, at the time, people selling, random CDs of, what we call pirated softwares, right? Of course, later we cracked down on that pirate software thing, correctly. But I think that was a really interesting time where, like you said, it's early days of computer internet, a lot of exciting things happening, but also a lot of what we can call chaotic scene, right? People buying random softwares, and
right.
But that was my childhood experience, and that's maybe lead to why I initially was studying electronic engineering when I go to college, because I always aspired to be a computer game designer. I was reading, 大众软件, People Software, and a lot of the early gaming magazines.
That was very popular gaming magazine in China
at the time. And then so that was my childhood. Basically, like everything I know was try to secretly, behind the back of my parents, playing games, maybe when they're away, or try to get on the internet without having too much too big a bill, and also trying to really from early days trying to be a computer game designer. That was my start.
It looks like you have such a fantastic child life, and you like the game design, and many kids also like the game design.
Yes.
And when you went to university, which is Tsinghua University, the #1 university in China, you someone have a different opinion. Top tier university in China. Yes. You end up choose biology as your career eventually.
Exactly.
Tell us like why you switched to do biology.
Yes. So I entered Tsinghua studying EE, electronic engineering. The two factors I think that influenced me. One is that I realized that electronic engineering department in universities are more focused on hardware rather than software. So I
made the exact same mistake. I was also EE when I enrolled my own university. Thought I was going to do software, right? But then it turns out everything is hardware related. Yeah.
Yes, and so so you will know. I was a little bit confused that we were mostly trying to learn how do you repair a TV, how do you circuits and stuff. So that's when that's one factor. It's like I kind of think that EE is not necessarily exactly the type of field that I want to be. But the second thing is when I was trying to think about, maybe I can change major, and thankfully Tsinghua allowed to do it. I had a personal change in my family where both my grandparents on my mother's side passed away in the span of a couple of months, unfortunately.
That must be a very tough time.
It was a very tough time because parents are busy in China. Being the only child, usually we kind of grow up with grandparents, right? Maybe even today. And so both of them were essentially like passed away because of fairly, common disease like diabetes. But when I asked around, no one seemed to really have a sense of why, they passed away so quickly. And as you know, the experience was shocking in the sense that, probably like, because I have no idea why people get, for example, diabetes, why people get cancer. But then, someone very close to me passed away so quickly from a disease that is probably my first experience, right? Because my parents were still very healthy at the time. Right. So I think that incredible.
That might raise a lot of questions in your mind.
And that raised a lot of questions like why we don't know, how people get sick and what are the ways we can really help people. And that's when I started to think about going to Peking Union Medical College or Xiehe, because I thought maybe I should take a career in medicine. But then, so the second part is really because family health, I shifted my focus to think a lot about how do we, tackle disease? How do we think about human health? And of course, I realized that being a doctor perhaps you save a few people. And perhaps by studying biomedical science, perhaps you can help a lot more people. And that's part of the reason I switched my major to basically biology and biotechnology in Tsinghua. Yeah.
Appreciate sharing the story. That is really, I know this will be a very personal story for you. The passaway of your grandparents had big impact about you, your worldview, and eventually lead you to switch to the biology. I think when they hear the achievement now you have, they will be very proud. Like,
I hope so, yes.
I wonder, you had this question on top of your mind when your parents passed away about learning how they got a disease, the root cause of the disease. Now that you have been in this field for decades and has advanced the frontier of research, do you feel that you get closer to the answers of those questions?
Yeah, great question. I thought two quick thoughts here is one, and maybe that can be something we expand later, is we are definitely getting a lot closer. As you know, right? When we were young, there's hardly anything that is actually effective of, say, preventing, people from getting obesity and helping with, in a very, very fundamental way, in some way reduce likelihood of getting diabetes. But now with Ozempic, right? We go with like all these GLP-1, like, from Eli Lilly and Novo Nordisk, like all these amazing sort of anti-obesity drug. We really have something that, right, when we were young, there's no such drug like actually reduce weight. Most of the drug at the time were like mostly like, more like snake oil, right? It's like you can claim they can reduce weight, but it might be just the honey water, right? Or random, herbal extracts, right? So I think we've learned so much about human health and disease that we are seeing these amazing, right, products that are helping people and even preventing, not just treating a disease, but even preventing people from getting, say, obese by using this GLP-1. So I think number one is definitely, I think we've made a lot of progress. But the second part, which leads to why I've shifted my whole research team really towards using AI and machine learning, is that even today, right, we still have no cure for type 1 diabetes. We still are far from saying, like, people are still need to take medications or insulin every week, take some insulin pumps, right? All this stuff. You have still a lot of disease that doesn't have any cure. It's more managing the symptom. And also, for Alzheimer's, right? There's still very big question, like what actually causes Alzheimer's? What causes aging? Even though aging is kind of a hot topic. So I think we're still far from really solving a lot of the, what we call complex disease. And even CRISPR, that we can talk later, is usually for very rare disease. So I think there's still a huge demand and huge gap, which partly to me is because we have very slow progress in terms of how we can actually make discovery. So I think the idea becomes there's a huge gap still in healthcare and discovery. And how can we really accelerate not just the progress of one thing, but maybe the progress of 100 things at the same time? It's basically like, right, when you have internet, people can build not just one thing, email. When you have internet, you can build 200 different websites. You have Google, Facebook, right? Everything, WeChat. So I think this is what we might need to answer the question you just sort of asked, which is how can we accelerate the whole discovery process rather than tackle one particular technology disease at a time?
Yeah, very well thought answers on that. Like, we have made a lot of progress in the past few decades to understand some of the diseases, but there's a lot more things we do, and AI can help with that.
Hopefully.
I would love to dive deeper later for that topic.
Totally.
But I want to go back to the early days of your life. And after graduating from Tsinghua, you came to the United States, and then you had many people would consider the golden ticket. You get a McKinsey offer. So you can go to make money and live a good life, live American dream. But instead you choose to go to academic research. Tell us some more. Why did you make that choice?
Yeah, thank you. Yeah, so, Yeah, I sometimes tell my students, right, and I think all of us agree that the decisions we made will probably has bigger influence in our life trajectory versus the, way that after we make the decision, how we work. So I think for me, some of the key decisions, like you said, is I was debating between taking a consulting job at McKinsey versus, continue academic research, in Boston. And ultimately, to be honest, it's because two simple things. One is much like I love playing computer games, I think I'm fundamentally driven by curiosity when I make decisions. I wanted to, in some way, maximize the sci-fi nature of my life in some way. So, or the gaming nature of my life. So I thought going to, consulting is fun and probably makes more money, to be honest, right? But then going to do research, if I can, I think it's really going to have more a unknown factor to it.
You had
exploratory factor. Yeah.
You had a great point. And the choice you made did lead you to one of the most significant scientific breakthroughs in the past few decades. You are one of the main researchers behind the technology called CRISPR. And in my view, is probably the top three most important technology in the biology in the past decade or so. Many people call it a genetic scissors.
Right.
How do you explain this technology for our audience who might have less background of biology?
Yeah, thank you. I think, I was very fortunate. And like you said, it's really because of the people I met. Like, going back to your early comments, like I met George Church and Feng Zhang when I was an early career, like student. And they really helped me to think about CRISPR. And that's how I can get started on that. And CRISPR basically is, we all know, like, there are 30 billion base pairs of DNA in every single one of our cell.
Yeah.
Right. And then it's almost like you can think about it as a Microsoft word processor, like, right? So in a way, our genome is a big 30 billion letter book.
Yeah.
And sometimes these things go wrong, or sometimes we wanted to, program or write a new book. So basically, to describe CRISPR, it's basically this very nice search tool that almost like a search and edit box in your Microsoft Word processor. It can quickly search to a specific words or sequence, in this case DNA sequence in the genome. And then once it recognizes the specific thing you are searching, it can make a cut, almost like an edit. So you can delete a few letters in the genome, basically a few DNA sequence in the genome, and then replace it or change it. So this is kind of what CRISPR does. And in a more specific way, CRISPR is basically like a very nice molecular scissor. So it's a protein and an RNA. And when the RNA bind to a specific sequence, almost like a GPS, it will locate to that specific sequence. And then the protein comes in as a scissor, almost like once the GPS locate this person, you deliver a cargo.
Yeah.
And then the protein basically then make a cut and essentially allow people to change the DNA in that specific part of the genome. So that's hopefully give you a sense of, basically it's like the whole process like editing a book, but then specifically it's almost like a little delivery truck that have a GPS, locate to the genome, and then deliver a cargo. But the cargo in this case is a scissor, like you said, that cuts the DNA.
You put it in such a visualizing ways, and there's a lot of similarity between us as a human and also the computers on that.
Right.
And also, I know that one of your main work contribution on the CRISPR field is that you prove that this technology can be done in mammals and eventually to humans gene editing. Can you tell us why that matters?
Yes, thank you. Fundamentally, I think there are three really exciting reasons for one motivated, my own work as well as of course George Church, Feng Zhang, and of course the Nobel laureates Jennifer Doudna, Emmanuel Chamete's work. and then it also show hopefully you the application of CRISPR. So the three quick thoughts are, number one, right? We have a lot of disease that's very much, genetically driven. Give you an example. Two very good examples are, there are hereditary disease, like, when a new child is born, they can be born with a simple DNA error, and then causes anemia. So I think if we can fix DNA, a lot of these inherited disease are going to be treated through precision gene editing. And then the other thing is a lot of risk that we have in disease. Like, for example, my grandparents have diabetes, so my whole family have a higher risk of diabetes. And there's also people have family risk of, for example, like short-sightedness, right? And then also like Alzheimer's. A lot of things are, but these are not simple. There are many risk factors contribute to this. If we can specifically, make cuts or perturb the genome, we can understand better what causes Alzheimer's, diabetes, and then hopefully find new drug targets. So I think these are what we can call like therapeutic application of CRISPR, right? That's one thing. And the second thing is agriculture, actually. So right now, if you go to supermarket in the US, and I think probably also in Japan, you can find potato chips that are actually using CRISPR engineered potato. And the reason because typical potato, if you move the potato from the farm to the factory making the chips, there will be a lot of potato that will be rotten, right? But when you CRISPR away some of the gene in potato that causes oxidation damage, that make it easier to have rotten tomato, you can make the potato less likely to become bad during the transportation. So a lot of potato farmers these days actually use CRISPR potato in the US. Anyway, so I think the other thing is like you can use CRISPR to really engineer plants, agriculture, farms that help improve our environment, improve our food, right? So that's the second thing. I think the last part is very interesting, and like stem cells aging, right? So this is like, instead of if you have say, oh, I have a disease, I'm sick, I need to CRISPR to help me, or I need CRISPR to help me find a drug, a target. What if you can improve our ability to live better, healthier, longer? Like for example, give you one specific example. Like some people, especially in Asia, right? When I drink alcohol, I become red on face, and this is because we lack some enzymes. If you have a safe way to give you that ability to take more alcohol, and sometimes you do have to drink, then you are healthier because you are less damaged by drinking alcohol. Anyway, so my point is like, if you can really nicely do CRISPR gene editing and help people, maybe there's also a way to improve healthy living by giving you ability to live healthier, to give ability to rejuvenate our stem cells for anti-aging purposes. So I think it's not just when you are sick you can use CRISPR, but when we are healthy we can extend that wellness through using some of the gene edit. So that's the three motivation for me personally why I really wanted to work on gene editing, CRISPR technology, and also why I think enable this technology in mammalian, in human cells will be very, very exciting. Yeah.
Wow, that's a lot of amazing applications. I think what like you mentioned, what leads us to live healthy, have better as humans, and have a better have less diseases, and also have better food.
Yes.
That's fantastic. You mentioned about you want to maximize the sci-fi part of your life, and now you go all the way into the core of the sci-fi.
Right. Hopefully.
It feels like the hands of the god, that it can change the gene, like genes and the living forms. That is great. I also want to help our audiences to understand how hard this technology is before we go to the details. Can you tell us, like, before CRISPR is invented, is there any way people can manipulate or editing genes, and how hard it before the CRISPR was invented?
Yeah, great question. It is very true that even before CRISPR, we can engineer the genome using technology like what we can call zinc finger nuclease tau nuclease, these are early technology that were able to already edit or engineer the genome. The reason why CRISPR is such a powerful technology, and give an analogy, right? If you think about why mobile phone is so exciting. When we were kids, right, we have this thing called this a big cell phone, 大哥大 in Chinese, right?
Yes, I
it's
remember
very expensive,
that.
very big. So
very big.
And then like a few people has it, but because of all the mobile phones, of course, iPhone, right, visionary, work from Steve Jobs and Apple makes the whole cell phone accessible, powerful. I think so basically like the early mobile phone sell for $10,000, and now it's like $100, right? You can get a phone or something, maybe five. I think that 100x reduction of cost makes things scale and impact. And then CRISPR, like you said, is not the first genetic technology, but before CRISPR, it cost someone $10,000 to make a single genetic protein. But with CRISPR, because it uses an elegant small RNA and protein to program the cells, you just need to synthesize a small piece of RNA. The whole cost of CRISPR a gene dropped to something like 10, even at most $100. So it's really not about necessarily if CRISPR is the first technology for editing. It's actually the first we can call scalable technology.
So the CRISPR really creates an industry.
Exactly.
Is the metaphor? Yeah, it scales. And I want to dive into the moment when you were in the frontier and create this technology, the CRISPR in the humans and the mammals. And you mentioned earlier that you went to the lab of Professor Zhang, at the Broad Institute. And back then you were a new student in the institute.
Yes.
And when did you start to hear about the CRISPR technology in your lab? And at what moment you started getting interested in this problem?
Yeah, so I think very quickly I met Feng Zhang in George Church Lab, but primarily, like you said, when Feng's become independent at MIT, I decided to move to Feng's lab to work on gene editing. And the first time I heard about CRISPR was really thanks to Feng picking up on some really inspiring study in bacteria, not at the time yet, in humans. It really, the bacteriologist, the microbiologist, initially discovered CRISPR in bacteria like E. coli, all these small organisms. And then because Feng and I have been chatting about gene editing for a long time at the time, so we decided, okay, if bacteria can use such a simple way to program their own sort of DNA genome, or basically like to essentially edit, engineer genome, DNA sequence, can we move that into human cells so that we can have, like you said, hopefully the first extremely simple, extremely scalable technology for editing genome of humans. So I think that's really got me excited. And like you said, the potential of this thing to scale, not just 2x, 10x, like 100 or even 1,000x is where I really immediately got so attracted to the idea that I started working on it with Feng.
Yeah, you put it a great way, and it actually reminds me of the invention of many great technology. It's not like when you're doing the scientific breakthrough, it's human just come up the idea from middle of nowhere. It oftentimes is coming from observations of the world around us. Right? Many scientists around the world will start to observe the bacteria. And in your cases, I wonder when you were doing your own research, are you aware of there are many groups in the world doing similar research?
Thank you. Yeah, so I think it's very exciting time at the moment. Back in the 2000, early 2010s, where like you said, I think initially really basic biology discovered this bacterial system to fight virus in bacteria. And we already had a sense that, probably other people might be working on the same idea, which has turned out to be true. And this, I think, like I said, is the theme in science. Like
Mm.
we can call it either great minds think alike, or it just really like the right timing will trigger, almost like will trigger the same, people have the same idea. I think at the time, we don't know exactly, for example, who are the other people working on the same idea, but definitely I think like you basically mentioned Jennifer, Emmanuel, who are the two Nobel laureates, their team are totally working on really thinking about how CRISPR, for example, can be used for human gene editing. And then even in George Church group, we have friends, who we didn't know at the time, but we later know that are, friends of us, in George Church group also were working on adapting CRISPR for human genetic. I think there are also Korean groups, even probably some group in China, in Europe for sure, like a sickness group in Europe also had basically around the same time trying the same thing. So I think in many ways, like you said, when the timing is correct, I think there will be convergence of ideas, sometimes even across the world. So I think, anyway, back to your question, I think we don't know exactly who are the people, but we had a sense that other people probably will be working on the same things. We also were working very hard and try to be at least, the first groups to show people that, yes, you can do human genome editing in such a simple, elegant, and scalable way using CRISPR.
As a student who were doing this research, will you feel time pressures? Will you had to work, like feel this urgency, had to work very hard to make it done ASAP?
Yeah, great question. Maybe I'll share two very quick stories around the time, right? Number one is that it's always easier to rationalize things afterwards. At the time, to be honest, I don't really know how long it's going to take and it was challenging. We had many failures. And I still remember, when we saw, for example, a pioneering work, primarily done, I think, in Emmanuel and Charpentier's lab, but also probably in collaboration, I think also with Dr. Jennifer Doudna at Berkeley. But, Emmanuel's team, with Jennifer, they have this 2011 work where they show that an important RNA processing step was important for CRISPR to work. So that's really enlightening. And I think, we really accelerated a lot of the efforts because of this very, very basic science work. And without that work, like I said, I think timing wasn't right, because you first need to at least know, for example, how CRISPR works in bacteria to some extent before you can actually use it in humans. So I think that early discovery work, is really critical, so that even though we were trying to rush things, a lot of times until the timing is right, even no matter how hard you work, you still are searching in the dark. So I think, but the first story basically tells us you can work hard, you can have urgency, but a lot of times right timing is still very key. Like if you don't know, the basic mechanism, no matter how hard you work, it's still not gonna give you the right answer. And the second quick story is that, I think we were in some way, overly, optimistic about that. We were maybe even if other people working on it, we might be still the leading group. But turns out, right, when we published our work, there were already other work, right, that were either publishing or to be published around the same time for showing human gene editing. So I think, to be honest, I think we should have worked harder and have more urgency. But turns out, because we didn't know who else are working on it, there wasn't like this, it wasn't like people would every day post your idea on the internet or Twitter, right, or Facebook. So we were probably less, hardworking than we should at the time. So which is fine, it's very hard to, it's always easier to say, oh, you should have done that afterwards. At the time, we didn't know. And then even to be honest, when I was in Feng's lab, there were up to 7 students, rotation students, others visiting students or people, who wanted to consider CRISPR and worked with me. But essentially, the first 6 or 7 people in the lab really did not necessarily always stayed in the project because they think, oh, it's too hard, too slow. So I think in many ways, right, even for me, I think it's very hard to tell how hard you should work. So it's really search in the dark. So I think in many ways, you just have to believe in the value and believe in what you think is true, and then try to play with it. I think a lot of times it's just really not easy to predict at the time.
Yeah, thanks for sharing us the real story and the real life of a scientist who is pushing the frontier. And it reminds a lot of my own life as I was doing research, not in biology, but AI side as well. And I would put it this way because many people would think they like this like dramatic stories about scientists racing to chase the Nobel Prize, that type of thing. That definitely happens when in more like industrial applications, when you kind of your goal is the goal, like you are trying to chase it. But the scientific discovery process is completely different.
Right.
The people who are in this game have absolutely no idea what they're needing to.
Right.
And in fact, that even if you like, they would work very very hard, but they are working with in a game that doesn't have objective, doesn't have like the finish line.
You can see like you're sort of searching in a dark, fog, looking for that little light that might come up someday, but you don't really know, when that's gonna happen, right?
Yeah, exactly. And also that reminds me, earlier you mentioned about the philosophy of life. It's never about optimize the things that you can see. It's about optimizing the curiosity and optimize about the sci-fi part of your life. In fact, that's also tied back to, I think that contributes to how you discover the CRISPR, because it's not about optimizing the final goal. It's actually try different things. And I'm very curious about like when you doing the CRISPR research, like have you tried other things that didn't work?
Yes, we have tried many things that didn't work. By the way, I have like 2 years of my PhD, right? So people thought that, I was probably very lucky, everything's smooth. I was working on CRISPR, published some really nice papers, and then, but actually almost 2 years of my PhD work never got published because I was trying to work on depression, schizophrenia. I was trying to actually use gene editing as a tool to study neuropsychiatric disease. And we were working, try to engineer the brain in the mice, but that was very slow and a lot of data was very noisy. Basically like you basically do the same experiment across 4 or 5 mice and the results can be totally different. So do you trust mice number 1 or do you trust mice number 3? Or maybe you trust none of them, just do it again. Anyway, so there's a lot of my work, actually days and nights of work in PhD that never got published. That's not at all related to CRISPR. It was neuroscience, psychiatric disease, trying to help people with depression, schizophrenia, which as we discussed, there are a lot of diseases that are very devastating, and we of course want to help them. But like you said, there are a lot of failures and probably more failure than success. But eventually, right, people mostly only see the success, which is fine. Like you said, maximize the experience, right? You cannot always maximize the goal.
Exactly. And you touched a great point about what is the right motivation for scientific breakthrough. If you optimize for success rate, like you mentioned, half your research doesn't need to meaningful thing. If like 90% of your research is going to be used or be successful, you're probably too risk averse or not push yourself enough. You're not really pushing the boundary of the scientific research. And you mentioned about you have tried so many things in the first 2 years. A lot of them don't work. Can you walk us through the particular moment you realize, oh, something is working? This might be something that is impactful. Is there any particular moment you realize, like eureka moment like that?
Yeah, good question. I think maybe there's less there is sort of the moment where, for example, we saw the first evidence of editing human genome back in the lab. That definitely was extremely exciting. I think the eureka moment is not so easy to define, because I think eventually what you really care about is, like you always needs to be believing in the things that you work on. So in some way, you always had expectation that one day it hopefully will work. Okay, so basically, so very quickly, my thought in this particular eureka moment is that almost like starting a business, right? It's sometimes less about whether you are the smartest person to work on a scientific problem or startup. It's more like, do you really believe in it so that you keep working on it? Right? I think if people, there's a very famous thing where like, right, in AI, which you're familiar with as well, because people like, some of the people like in OpenAI believe in the scaling law, and many other people don't believe it. It's not necessarily true that OpenAI have the smartest people at the time. The reason why ChatGPT or GPT is such an amazing breakthrough is because they were just keep working on it.
Exactly.
Basically, like, they believe that scaling law exists. And at some point, if you make the model bigger, learn more data, train with more data, it will become very, very powerful, intelligent, which is turned out to be true.
Yeah.
I think so. I really think it's not about a specific, I'm smarter than other people, I have this super. It's really about, do you believe CRISPR would ever work?
Right.
That's the difference.
Yeah, you put it in a very great way, which is like many people think scientific breakthrough is like, there's a eureka moment, there's a particular moment this idea has been invented. But in real world, it is a gradual process that you keep pushing forward. And so you have to have the belief, like in your case, it's CRISPR. It's like years of work put into that, gradually improving.
Yeah, I think that's the number one key.
Exactly, yeah. That's the end of part 1 of our conversation with Professor Cong. In part 2, he takes us from CRISPR to the frontier of AI for science, sharing his vision of AI co-pilots, autonomous scientists, and multimodal models that could transform biology and medicine. Don't miss it. Subscribe to FounderCoHo if you haven't already.